from monoclonal antibodies to chimeric antigen receptors for the treatment of human malignancies. Monoclonal antibodies and their directly drive cell-based application known as chimeric antigen receptors ensue from the need to develop novel therapeutic strategies that retain high anti-tumor activity but carry reduced toxicity, toxicity compared to conventional chemo and radiotherapies. In this concise review article, we will summarize the application of antibody, antibodies designed to target antigens expressed by tumor cells and the transition from these antibodies to the generation of CARs. The use of MAB in cancer patients was pioneered in 1980 by Nadler et al. In patients with relapsed lymphoma. 12 years later, the FDA approved the first therapeutic monoclonal antibody, orthogonal OKT3, for the treatment of patients with acute transplant rejection. In the last 30 years, more than a dozen of MAP specific for different antigen of interest for human malignancies have been developed, received regulatory approval, and marketed. Although received anti-CD20 MAP is the most extensively used, over 2,900 clinical trials based on the administration of MAB have been reported, and many others are currently ongoing in cancer patients. In addition, the development of MAB experienced a significant technical evolution from the first production of murine, rabbit, and chimeric MAB into the new generation of humanized MAB that leads to lower immunogenicity and therefore longer life in vivo. These therapeutic maps are designed to recognize a tumor antigen and cause death through various mechanisms. Briefly, maps can directly induce apoptosis of tumor cells or directly eliminate them by recruiting various immune cells with cytotoxic properties, um, or by activating the complement cascade. In addition, MABs have been modified to carry tumoricidal substances such as chemotherapeutic agents, radioisotopes, toxin, or small molecule. Some of these antibodies, such as SGN35, have been approved by the FDA and are in use for the development of CD30 Hodgkin lymphoma. Finally, a new group of MABs called bispecific T cells enhancing BITE antibodies have been developed to simultaneously target two antigens, one on tumor cells and the other one on effective T cells, to recruit and colonize immunological activity cells to the tumor. What is a BITE antibody? Bispecific T cell engagers are a class of artificial bispecific monoclonal antibodies that are investigated for the use as anti-cancer drugs. They direct a host immune system, more specifically T cells. Oh no.
Cancer research is progressing more rapidly than ever. The number of oncology drugs approved by regulatory authorities has substantially increased over the last few years. One such area of research continuing to deliver breakthrough innovations is immuno-oncology, designed to harness the body's own immune system to fight cancer. Now we are beginning to see immuno-oncology therapies that specifically harness the power of the T cell, the central cell of the immune system. Amgen is leading the development of numerous therapies that specifically engage the T cell. Amgen developed the first bispecific immuno-oncology program using a BiTE, or bispecific T-cell engager, that can be engineered to target tumor antigens expressed by various types of cancer. BiTEs are off-the-shelf therapies because they do not depend on individualized manufacturing. You gotta tell us that. The true challenge in cancer treatment is often not the therapy after first diagnosis, but how to prevent relapses that often occur despite treatment. Relapse is attributed to the lingering presence of cancerous cells, even if the majority of the cancer is eliminated. Efforts to eliminate remaining detectable cancer cells are an important area of investigation for biotechnology. BiTEs are protein high in vitro more than a of cancer. We are also studying next generation and ever as own oncology. Amgen is leading the development of numerous therapies that specifically engage the T cell. Amgen developed the first bispecific immuno-oncology tumor and depend on diagnosis, but thousands of cancerous cells are an important two T cell in the body translate more patient bites, which fit across a variety of cancer types.
Immunotherapy that utilizes the body's immune system against tumor is a promising field of cancer research. In recent years, exciting technologies and platforms have been developed for the intricate design and production of anti-cancer immune reagents using antibodies in the form of single chain variable fragment. The SCFV technology has been adopted and used in some promising cancer immunotherapies, including bispecific T cells engagers and chimeric antigen receptor. Both strategies use SCFV to direct cytotoxic T lymphocytes to specific surface antigens on cancer cells to facilitate a polyclonal T cell response to tumor antigens. A BiTE is a recombinant bispecific protein that has two linked SCFVs from two different antibodies, one targeting a cell surface molecule on T cells and the other targeting antigen on the surface of malignant cells. The two SCFVs are linked together by a short, flexible linker by binding to tumor antigen T cells simultaneously by mediate T cells responses and killing of tumor cells. Uh oh.